properly. So you can just download it from online. You open this R file, the software, and you see this ROM file. You can just drag it inside this R program for it to run. And the first thing it tells you is to select ROM.R file again. This is to basically locate the folder where you have your R files. So I have it pinned here, and you go to run and just click on open, and now it's processing. So at the first time when you run it, it's going to download a lot of library, because this isn't my first time running it, so it's not really downloading anything. It's just going through the list of code I have. But then when it starts to install things the first time, don't worry, that is normal. So it asks you where the results should be stored, and you can just find a location that best works for you. I have my own location here, which is inside this folder, in the test folder, and I'll just put it right here. Click on OK. And there are two options. You can choose a file directly from XQuant, or if you choose other, there's a certain format. And so this pipeline is usually for processing phosphoproteomics. But if you have a proteomics data, you can still process it via this other option. I can show an input from a normal peptide proteomics peptide file instead of a phospho file. So this is basically, so the first 6 columns are annotations, names and things, and then all the rest are data columns. So normally if you have a phosphoproteomics data set, you would have all of these 3 not in NA, so they would have value. However, if you only have your proteomics data, you can just enter NA for these sets, and the pipeline would know this is a proteomics set, and it will proceed normally. It wouldn't give an error. And I guess this time I will just show a regular phosphoproteomics data. So I will choose XQuant, and it's from XQuant output. So I click on OK, and it's asking me where is the result file. So I would just go into the folder that has my result file. Let me see if I remember where it is. I made a short one out of this from just random row selections. So this is faster to run. And I click on OK. It asks you if you want to remove rows with too many missing values. I guess in this case, I'll just say yes, just to show what will happen after that. But you could choose you're not removing any rows either. So if I click on yes, it's going to ask me for a cutoff. And right now the cutoff is set for 50, 30, and 10. So 10% is the most stringent. If you choose 10%, then all the rows that has more than 10% missing value will be basically removed, filtered out. I usually choose just the 50%, and I click on OK. And now it's asking you if you want to remove samples. So because sometimes they're outlier samples, it's better to remove them before analysis. So yeah, analysis, the result of the analysis would be more correct. But however, like time series for this kind of data, for example, that you shouldn't remove any samples. So I have over here, probably you should be careful with removing samples. I will just say yes for this one. And then it's asking you to choose an organism here. And then for this data, it was our neuron culture, so it was from mouse. And then for normalization and imputation, you can choose neither. So I've been choosing neither for our recent data set because I didn't want to change the data. However, you can also choose normalize or both normalize and impute. For this imputation method, it requires normalization for your data. So for this one, the normalization is quantile normalization, but for only normalize, it's actually medium normalization. I guess I'll just do this one just to show the functionalities. So for the analysis steps, I'll choose all steps to show every step, I guess, from the pipeline. If I click on OK, now it will ask to set up the groups, and I actually have this very wordy explanation inside the pipeline. I usually just click on no because it's really wordy, but I can just show what it says right here. So it explains how you input your groupings, and this is basically an example over here, how you write different groups in what way, and then also it's recommended that you look into, because I extracted all the samples in this file already here through the pipeline. It's good to look at this to set up this number, because even if you have a list from before, now after the sample filtering, so the sample may not match up with your original files because some sample might be removed if you've chosen sample filtering. So it's good to look at this file here. So for this one, it's actually using only one way to group the data. So you have 4 groups, and each group is has a different genotype and treatment over here. So I will just set this up just to show how to set it up. And this will be the list of samples. So you set it up based on your sample labels. So click on OK here, and it asks you how many ways can the sample be grouped. So for the preparation here, I will say 1 way, and then it asks you a list. So how is this grouped by? Because it's by both genotype and treatment, so I'll just click on others and type in genotype and treatment. You can actually type in anything here. It's just for you to keep a note of what this comparison is, or this group is. So I click on OK, and it's gonna ask me to enter group orders of group 1. And one thing I guess to note is that for the fold change, I set it up to have the higher number dividing the lower number. So that would be the order. And usually I think it's KO divided by wild type. That's easiest to sort of see the change, and also treatment comparing to not treatment. So when you're setting up the order, you kind of look at the normal type and untreated first. So this will be number 1. And then you go to the next one. What I did here, number 2, is actually keeping the treatment to untreated, but change the genotype. And then group 3 and group 4 in the same kind of order. So I have a list here already. If you can see, they kind of match. So this is KO, and this is DMSO. And so over here, it's group 2 for KO and DMSO. So this is a 2, you enter a 2, and then you have a comma, and then you go to the next one, and then you check the groups here, and you just enter the number. So I have a list here already. I will just copy and paste the whole list for the group inputs, and I enter it here, and I click on OK. And then it's gonna give me some more instructions. This is for group comparison. So it basically gives you instructions on how to enter comparison information, which I will show right here. So there's more instructions. If you click on no in the beginning for instructions, they won't pop up, these wordy instructions. Okay, so it asks you which group to compare. So there would be more lists over here if you have more than one way to group your samples, but we only have one way, so we'll just have to choose from here. So we will compare groups from this way. You click on OK, and so you can compare basically 2 or more treatments. And so if we want to compare, let's say, wild type versus knockout for both non-treated, you can enter 1, 2. You can enter in whatever order you like. You can enter 2, 1 as well. The fold change will always be the same. It will always be 2 divided by 1, group 2 divided by group 1. And I can say this 1 compared to 2. I click on OK. And it's going to ask me if I would like to make another comparison. If I click on OK, and it's going to show me the same window again. I will choose the same one. There's only one for this one. And then this time I will compare 1 and 3, which is comparing the different treatments. And I click on OK. It's asking me. So it will keep on asking until I click on cancel. And then if I click on yes, and then I choose, let's say 3 groups to compare, let's say 1, 2, and 3. So for this, it will use ANOVA directly. It will figure out on its own. And so if you plan to compare more than 2 groups in your comparison, it's good to, in the beginning, to choose normalize and impute for your imputation options. We'll show this like 3 comparison as well, I guess, here. So I'll just choose 3. So I will have these comparisons, and now I will click on cancel. So they will just make these comparisons and no more. So now it goes back to imputation. So everything is set up. So now it's going to perform the imputation. You can actually take group label into consideration for imputation. And so for this one, I can click on yes. So let's say if you have missing values in one of the labels, and it will take your group label into consideration. So it will actually align that to this group that has a missing value. So it doesn't look at the whole data, it looks at this specific group, I think. So I click on yes, and it's going to ask me which group labels to take into consideration, and it's still there's one kind of grouping, so I'll choose this one. Just click on OK, and now it's actually doing the imputation step, and then that's done. So the pipeline, the first step of the pipeline is actually overview figures. So it will directly go to overview figures, and then I click on yes. And then because there are colors for the figures, it's asking you how to color your different groups. So there's only one grouping, so I can only choose this one. Click on OK. Now it's gonna just keep on running, and this is one of the figures making it.
So if the figure seems to be cut off, is that because we can't see the entire window, or because it's that is everything we're seeing?
Because you can't see the window, I can show the actual result in this window. All that is running here. So now you have more group information here. You have the comparison information, and then you have the sample order that we just did over here. So this is the result folder. So now there's the overview figure file, and the cube that you were seeing is here. So you can access it whenever you want. It's in your result. And the 1, 2, 3, 4 label is matches with these groups. Then now now this is the statistical analysis step. So the first thing it asks you is the data pairs. This is asking you for the first comparison. Since our whole data is not paired, I'll just say no. And so this is for 2 group comparisons. You have these tests to choose from. You can choose any one. You can choose more than one as well. You can choose any of them actually. So I guess for this one, I'll just choose everything. So it's going to basically perform all these tests. And I click on OK. And so this one is for the second comparison. And since the data is not paired, I say no. And it's also a 2-group comparison. And just, I guess, choose everything again, or just maybe these.
So the data is now log2 normalized. Transformed.
Yes, before it goes through this test, it will be log2 transformed for these. And then this is for the ANOVA, and it will just perform ANOVA for this comparison because it was 3 group, but it also asks if the data is paired. So I'll choose no for this. And then over here, this is the warning message. Make sure that the normalization imputation option was selected for valid ANOVA result. It will always have this pop up just to remind users. And then now it's doing the analysis here, and it's gonna take a little bit to run everything. Before this is ready, I can, I guess, show the raw data here. The information has all of the raw information versus also the intensity, where the intensity comes from. So this is different sites because Max Quant also gives intensity for each number of sites. So if you have this peptide and this phosphorylation is on this peptide, there might be other phosphorylations on the same peptide, and it gives you either there are no other phosphorylations, one other phosphorylations, or two other phosphorylations, and the intensity for each of these different types. And so over here, it has a percentage of where it comes from, and some of them are NA. This might be just 0 values, and then most of them is 100% in one of them. But then sometimes you can see, like over here, it's mixed. It's from these two different kind of peptide with different number of phosphorylations. So you can actually check this to see where it's coming. Okay, this is the pop-up window that I was talking about. It will pop up, and if this one was not responding, then this will come up to be all empty. You couldn't really see anything. So this is choosing a cutoff for the first comparison, and you can choose again all of them or just one of them. Let's say if I choose fold change and RAS p-value here, then I click on OK, and I would choose a cutoff. I just choose the regular one, the 2 and the 0.05, and now it's gonna give you the number of significant entries. So and it's gonna ask you if you want to keep this significant list, because sometimes the number comes up to be 0. Then in that case, I will not keep the list. So it's like it would never, it never happened, basically. So there's 13 significant ones. I will just click on keep. And now it's going to ask you if you want to choose another set of cutoff for comparison 1. If I choose yes, I can also like look at, let's say, the overlaps of two tests. Let's say these two overlap. I just select these two, and I choose, let's say, 0.5 for both of them. Then it's going to give me the overlap of them that's significant. So there are also 12 for this overlap of these two. And then I can click on keep the list or not. I would just not keep the list for this one. So it's just to check. And then if you want to choose, let's say, fold change versus another like rank prop over here, you can just type in your cutoff, and it will tell you like there's 23 significant entries for this one. This is normal because I made a very small data set from the large data set, so there's like a lot less significant entries. So I click on yes. It's going to ask you if I want to set another cutoff for comparison 1. But if you're done with choosing your cutoff for comparison 1, you can just say no, and then it says it will move on to comparison 2, and then so for this one, I'll just, I guess, choose the same buffs, just to be fast. Say yes, and another one. Choose this one. And then keep, and then I'll just go on to comparison 3. for comparison 3, it's ANOVA. So you have all the 2 key ones as well, versus also the ANOVA p-value and FPR value. So if you just want to see one of them, let's say 1 versus 2, so comparing the genotypes, then you can just choose these fold change and p-value cutoffs and choose fold change for 2 and p-value for 0.05. It's going to tell you there's 12 significant entries. You can click on OK. And if you like to choose another one, it's going to show you the same window. Let's say if you want to have ANOVA p-value to be significant, it probably is, and then the fold change and this p-value significant as well. So an overlap between these, you can choose all 3 and click on OK. It's going to ask you for your ANOVA p-value, and then it's going to ask you for a fold change for this comparison, and then it's going to ask you a fold change, a p-value again. Then you set all 3, and then you see the number of significance. I think this is less than 10, so I'm just gonna discard this because a lot of analysis like enrichments for 7 significant entries, it doesn't work well. I'll just discard this, so I guess. Then you can choose, you're not gonna select another set of cutoffs, then you can just click on no. And then it will tell you this is done, and it's gonna move on to volcano plots. So for volcano plots, you click on yes, and then it will show you all of the groups over here. And for volcano plots, you can select any number of these comparisons. But it's going to ask me what kind of cutoffs do I choose for the volcano plots. I'll just choose the p-value one. It would ask me about this fold change and p-value cutoff. I have these 4 different choices. I think for the next version, I can let user to choose their cutoffs, but for now, I will just choose the 2 and 0.05. So 2 fold change and 0.05 p-value for the significance for the volcano plot. And I click on this volcano plot. So it's here. And so for the labels to appear, each side has to have less than 30 significant values, because sometimes label doesn't show up when there's too many overlaps. So I set a hard cutoff. These labels only show up if there's less significant hits. And then this is comparison 1, comparison 2, and there's just 2. So now I will continue with the annotation step. So now it's gonna go to annotation. This step actually takes the longest time because I use one of the packages. I think it gives really good information. It has a really long running time. So I think for this demonstration, I will remove that step. So now it's in annotation step. I have this warning, it can take very long, so you can leave it overnight. And if you click on OK, it's going to ask you. So this is the step that takes a very long time to get uniprot information. So I'll just say no for this one, for this data. And then human homolog information, then I'll just say yes.
Where is it getting that from you?
This is getting it from the ensemble data,
Okay.
this database. So everything is from there. And then now it's running this step, and so this is, you have your whole result from the statistical analysis, and all the tests you do is at the end over here. Beginning, you have your data, and you have your annotation here. So this is the whole set, and then for this data, I only had very few rows of data. So when you go to your significant list, these ones are the significant ones from your cutoff that you set. So if you go into one, it gives you this list, and this will be the cutoff fold change, and this will be the cutoff p-value over here. And this is the identity. And actually, when the pipeline goes on to enrichment step and also network step, the list they use is from the significant list. So for the enrichment, unless it's this kinase GC. GC is for the entire data set, but then for the enrichment analysis, it looks at the significant sets to see if there's any enrichment. It's gonna pick up these files and check these enrichment, and it's gonna do the enrichment analysis and network analysis for every single list here. So if you have a list that you didn't want to use, from the beginning choosing part, you can just say no, I'm not choosing it, because all of them will be analyzed later. And it's still running. It does take a while. But the folder is here already. The annotations, so the human homolog here information is extracted. I'm thinking it's working on this one, alignment. So all of these parts annotation is actually for your own organism. It extracts this information, and then it extracts the reviewed site information, and then the entrance ID, and then it extracts also the chromosome position too for your protein. And then it goes on to, so all the columns that starts with H. sapiens, these are the human homolog information, and it gives you gene name, it gives you so different things. Like I think it extracts actually everything from ensemble that's human related homolog information, and you can actually see this is the identity comparing these two proteins together, and you can see these are quite high identity. Well, this one doesn't, but usually for mice and human, you can see a quite high homology identity for these proteins. You can just kind of pass through it. And human extension is over here. These extensions, some of them you can't get it, but all the other ones. And then so it's still running. It's running this alignment. So there's protein sequence and protein, the coding sequence for proteins. So this is actually only the coding sequence, but then it's in DNA sequence. So it matches exactly with the protein sequence. It doesn't include exons or any other codes over here. So if you go to the protein sequence, you can look through the list over here. If you want to see the alignment for human homolog to your own protein, it's aligned right here. Just get anyone. So this identity is 91%. So this one serves as a reference. If you're looking at a site on mouse protein, you can kind of use this as a reference to check the human position.
Yes.
The thing is, like, this is a 2D alignment. It should be pretty good with this blossom matrix. However, like 3D alignment would be the most accurate alignment. So this is not 3D alignment, but I think it could, especially when the similarity is high, it could serve as a pretty good reference for checking the position in humans. Then this file is the same results. However, in the end, it has basically a reviewed list. So over here, it says if the extension is reviewed. So these are the protein extensions. Reviewed extensions is the Swiss Pro, I think. So all of them have been manually, like, sort of confirmed. Oh, this step is ready. I can set up the next step. So this is going to enrichment, and if you just click on OK, it should have done everything on its own. So there's right, no, you don't have to make any choices here. So it's just performing these. So for this one, sometimes when they're not reviewed, you look into a reference database. The site position that they listed is from actually the same protein, but the sequence is from a reviewed proteins sequence, extension sequence. So it doesn't match up when you search on a database, the site positions. So over here, it tells you if your extension is reviewed, and a lot of them are not reviewed, and then it extracts the reviewed extension here for you, and then it extracts so the position within the reviewed extension, the same position that you're looking for. So if you search for these sites, then most likely you'll find it in your database information.
I have one question. So
Yes.
sometimes your site can appear only on an isoform. So is this mapping isoforms as well? So if there's no ambiguity about the site being on an isoform, will it try to map to an isoform? That's the question.
I think it might not, because actually it doesn't recognize isoform, because isoform, you have this extension, then you have this extension,
Yes.
and you have a number, right? It never actually takes this. It doesn't recognize this. So whenever I actually have to do this, I have to remove the isoform. So I had to actually literally change it. I have a cleaning step for that, so it's really unfortunate that I couldn't really do that for isoforms.
Is
It
it
doesn't.
possible to have some kind of warning column about this?
Yes, I think that's a very good idea. I will make a warning column to say that this is isoform, whether it's isoform. I think, I guess. So now it's running the enrichment step, and I guess it's done these two. It should be done soon, I think. So for cake enrichment, it's going through every comparison here, but when the comparison doesn't have any significant enrichment, it won't have information here. So all the information here are the ones that actually have significance. So this is comparison 1 out of 2. And this list, you can find it within the, I think here, the statistical analysis significant list. And there's the comparison 1 cutoff 2. So you can see which protein, which entries are in this list. So this is the actual list that has 24 different sites, I guess. So these are the proteins that's here. Oh, yes, I'll close this one. And you can kind of see these are extracted from that list. I think the count is a bit low.
Yeah.
I think now it's continuing to run these 2 steps. So they are specifically for peptides. Okay, now it pops up. So kinase analysis is specific for phosphoproteomics data. So for proteomics data, these 2 steps are not performed because you don't have site information, and it would just go on and do this. It's gonna ask you because for this kinase analysis to work, they want significant sets. So it's asking you for a p-value to basically have a cutoff, significance cutoff. So like, let's say I'll just choose RAS, then you click on OK, it's gonna just work on that. What it does is basically first it matches your sets of proteins to phosphosites that might be phosphorylating this site. So it checks a database which I downloaded from Phosphosite Plus. it checks all the kinases available and checks their substrates, and there's a calculation for which substrates might be phosphorylated by which kinase, and then it checks which of your peptides are significant, and then after that, it actually gets the fold change of all the significant peptides. It doesn't exactly look at the fold change, however, it looks at the direction of the fold change to see the kinase direction. So even if it's a very low fold change, like a minus 1.02, it still counts as minus. It still counts as like decrease in phosphorylation. So in that sense, but then it does only choose the significant ones when there is a significant test performed. Let's do that. Then I think that's done already. So now this is the last step, which is the network analysis. And so now it's gonna, this download happens every single round because it's gonna download information for your protein list from the string database. And then it's gonna do, it's gonna show these interactions specifically for your set only. And it asks you if you want to fund hubs, because this takes quite long. So I have this option here. You can say no if you're in a hurry. I'll just say yes, and then it asks you what kind of hubs, how you choose a hub cutoff. I usually just choose the first one standard deviation above the mean. That usually have like a good set, or you can choose the top 10 highest interaction ones. Both are okay, I think. And then it asks you if you want to do this interaction enrichment, which is generating 1,000 background for every hub and check the significance. Right, and then I just say yes to do that. But you can say no, then it's not going to do that to save time. So while it's running, I guess I could show other results. So that was the enrichment that we saw. So now there's, well, SoundMaker. A lot of times this one doesn't show up because it doesn't have good enrichment. Like sometimes it comes up empty, so this folder wouldn't show up. But when there is something shows up, it's here. However, sometimes when you have one count, it's usually not very related to your data because it's only one peptide overlapping. So when there's more overlapping, it might be like more relevant for this one. I guess new one, it's still relevant. That's interesting. So you can go on and just check the gene that's overlapping, but it's in gene ID. So this, if you enter gene and enter this ID in a Google search, it'll probably show you the name of this gene ID. So exactly what is overlapping from our data set and this cell marker set. So now that the pipeline is officially done running and everything should be in your result list. This is the whole thing that you get, and so the enrichment. This part is also only done for phosphoproteomics because it's phosphosite enrichment, and this is using actually just one, not a package, but one kind of R software to do. And I use also their database, which I thought was, it looks nicely extracted, most of it, I think, from Phosphosite Plus. These are only input files over here. I have to make special input files for this to work. The results are all inside here. So your comparison 1, comparison 2, and comparison 3 each are in a different folder. And then if you go in, you will have this result here. Then you will have your rank plot and your rank plot here, and you can check these. And then I think because usually if you look at fold change, it makes more sense than p-value because this p-value, like the direction it goes is different than the fold change, and it's another set of meanings. So usually if you only look at the fold change one, then it makes more sense. And over here, of course, it's the p-value are not significant at all, and it's not really changing in one direction, but sometimes it does change in one particular direction, and when it does, it's quite obvious, and it looked like good results if it does.
So those are, that's based on motifs, right?
These are based on peptide signature sets. So they have a signature set that has
yes.
some.
Now I got it. Yeah. Yes. Sorry, I asked you this before. Does everybody understand that? No questions. Yeah, they must understand. Yes. And of course, we can always look up this GSEA database to learn more,
right.
or you might have a file there with some information on it. Yeah.
Yes, actually, it's in my pipeline file. These are the database that they use. So actually,
really nice.
yeah, I think you can just go in there and see which peptide they include. So say for this one, this list, there's only 5 sets here, I think, and then they would actually write it in this way, pretty cryptic, and then you have each individual site here that has actually the window. So in the middle, it's the phosphorylation, and then they give you this window here.
Right. Oh, yes.
So this is the enrichment, and then this is the kinase analysis, and I have all the significant kinase extracted in this motif form, and you can see the kinase. So this is like supposed to be significantly changing in comparison 1, and then in comparison 2, this is supposedly significantly changing. And then I think it was just the scores here, and there's a earlier score one. So from here, if you see that the P greater, which is increasing in activity, if these are significant, then these kinases are supposedly increasing activity, and you go down here, you check the P less, and for these ones, they're supposed to be decreasing in activity over here. That's significant over here. So, and then network, this is entirely done on string database now, that the other database doesn't. Oh, these are actually done on SwingR results. So from kinase analysis, I made these networks that actually have all the information included in one figure. The thing is that the labels are really hard to see, I think, but they're there if you want to just check. So for for this protein, you have these 2 sites that's phosphorylated, and you just check on these interaction edges over here. Then you can trace it back to the kinase that phosphorylated it, like this purple one. You can trace it right back to here, and then you can see the identity of this kinase that might be phosphorylating it, which is this one.
Okay, see beta. Aha. That's brilliant.
And because the lists, significant lists, really didn't have enough proteins, so I think the network doesn't really have enough connections for it to reach a point where they can have a hub. So the hub analysis is actually not done for this when there's no interaction. But I'll just I guess explain a few things. So these nodes are the peptide or like the protein set of your significant peptides, and these edges are the interactions between them. And I only I set a cutoff for these interactions. It's everything above 0.4 in confidence level, which is also the default confidence level for string database. String database define high confidence to be a confidence level of over 0.7. So I also have that actually for my kinase information, but I don't have it on here. So everything that shows up, these interaction that shows up has a confidence level of 0.4 or higher. So the confidence level go from 0 to 1.
Are these physical interactions or gene
Actually,
expression interactions?
I think it has everything. 'Cause I don't think I
Oh,
have
everything combined.
filtered on that. Because there's many, because it's done on different database, I think, or different sets, and then there are different colors over
Oh, I
here
see.
that's
Okay.
from different sets, right? So I don't actually know how they define different sets.
So then if one goes to the string networking, they will tell what these colors mean, right?
I would think
Which is
so.
like- yeah.
I would think they should.
That's great.
And also, I actually, for calculation purposes, for the hub, I combine all of these into one. So it's either these two are interacting or these two are not interacting. So if they are interacting, then they have at least one of these interactions having a confidence of 0.4 or above. So let's counted it that way. And then the color outside shows actually the fold change from our own data. So the red one is increasing, and it doesn't have green one here, but green one would be decreasing. And then you have a not colored one, but then if you want to see the color one, sometimes it will cover your proteins. So I also have the options of not showing it, but then you have the color here if you want to see it. And I think that's about all.